Hello, and welcome back to Introduction to Genetics and Evolution. We've talked in some of the previous videos about mutations and about some of the complications beyond standard simple single gene inheritance. What I'd like to do now is come back to mutations just briefly, talk about mutation rates, or essentially how often do mutations happen. And I mentioned briefly previously that they happen a lot, but let's actually put some numbers on this. Well, again, let me reemphasize, all genetic variations started as new mutations. Now, there are two questions people often want to know. There's actually a third one that we won't talk about, which is how often good mutations arise. But people often want to know, how often do any sort of new mutations arise? How often is there any sort of alteration to the genetic code? Like, do you, for example, have any mutations relative to your parents? Will your kids have mutations relative to you? And then related to that, how often do bad new mutations arise? Like, do you have any bad mutations relative to your parents? Or will your kids have some bad mutations relative to you? Theoretically, we could do the same thing with good new mutations, but they're so infrequent and hard to measure that we probably can't do that very easily in this context. Now, how do you measure a mutation rate? So how do you measure a rate of new mutations? Well, mutation rate is typically measured as per generation and often per base. But again, measuring this, you have to define your parameters very clearly. You have to standardize by unit of time, which is typically the per generation. And you have to identify what kinds of mutations count. Like, are you identifying the mutation rate from one specific base to another specific base, like the mutation rate of a particular site from A to G in particular? Or do you mean just from that base to any different base, from A to anything but A? So this would include a mutation to G, a mutation to T, or a mutation to C. The other thing, too, we've often talked about this in the context of DNA sequence, but there are other kinds of genetic mutations, too. There are things like inversions. This is when you have a stretch on the chromosome flip, literally like just completely reverse its order. So all the same nucleotide sequence is there, but the order of it has changed. Or translocation, something where a piece goes from one chromosome to another. Or deletions, just a segment of DNA that just gets erased out and then the two ends get stuck together. Do you count those or do you not count those? And what people traditionally did, if you go further back in time, is people would measure mutation rates in the context of anything in a gene that yields a phenotype. Now, this, of course, ignores a lot of neutral changes and things like that. So you have to define what you're talking about. Typically speaking, people tend to focus on this when they're looking at a mutation rate, from a specific base to any different base. So let's say, for example, that you know it at one particular site, you started with an A, that when it changes to something but that. Okay? So we'll use that for the rest of this lecture. So how do you get this mutation rate per generation? Well, again, as I said, we'll use this base to any other base definition. And what people often do is they'll get a sequence of either a whole genome after a known number of generations or a big stretch of DNA. So let's imagine this is a 10-base genome. This is obviously ridiculously small. ACA, GG, CATTT. Okay, and after one generation, you look at the offspring and it's ACA, GG, CAT, CT. So what do we have here? We have one mutation, right? How many bases do we look at? Well, we looked at 10 bases. And how many generations? We looked at one generation. So in this case, the mutation rate would be 0.1 mutations per base pair per generation. So that's the kind of thing you can do. This is something that's commonly done in model organisms, such as, for example, C. elegans. This is a classic worm example. Now, another thing people will do is to compare sequences from existing species and estimate the number of generations. So, for example, if you could get a guesstimate of how many generations there are that distinguish humans from chimps, and you counted all the base differences between humans and chimps, you could get a mutation rate from that as well. So let me give you a couple of real numbers. This is an estimate of a mutation rate from worms for these C. elegans model systems. So it's 2.1 times 10 to the minus 8. This sounds very small, but trust me, it's bigger than it seems. 10 to the minus 8 mutations per base per generation. Now, what would this actually mean if you apply this mutation rate to humans? Well, the human genome size is 3.1 times 10 to the 9 bases. So how many mutations, if you just extrapolate directly from this mutation rate to this genome size, how many mutations on average would you expect individuals to get in one generation relative to their parents? The answer from this is 65. So that is not a small number of new mutations, right? And as I said, mutations are happening all the time. And amazingly, this finding is absolutely confirmed. That you see a 2012 study from various Icelandic families found on average 63 new mutations. This shows the value of extrapolating from model system studies. That estimate of 65 came ultimately from the studies of C. elegans worms. And from that, I just did some math to calculate what we would expect to see in humans. And in fact, we see a very, very, very close number to what I predicted from the C. elegans studies in humans. Now, interestingly, I didn't mention this before, there's a very strong effect of father's age. Now, most new mutations that humans get tend to be from their fathers rather than from their mothers. The reason for that is because in the production of sperm, you have many more cell divisions than you do in the production of eggs. So there's many more opportunities for an error to come in or for a mutation to come in. And in fact, what you see looking at this, there's a very strong age effect of the father. So here on the x-axis is the age of the father at the time of conceiving the child. On the y-axis is the number of new mutations found in that child. And what you see is there's a very dramatic increase as the father goes from 20, 25, 30, 35, 40, you see way, way more new mutations in the kids. In fact, for every year of the father's age, you see, on average, about 2 more new mutations. And this holds up with disease studies, that people see higher incidence of diseases like autism and other things like that in kids from older fathers. So this is something to consider, and it has definitely societal implications as well. So, coming back to what I said before, your brother is a mutant, but sadly, so are you. So you can call him a mutant, but he can call you one as well. But very importantly, these new mutations arise and they introduce a lot of variation into natural populations. And this ties back in with the previous video I just emphasized to you. Now again, what happens when these new mutations come up? If it's a new mutation, it's not been found before, then you have the birth of a SNP. You have this new sort of variation out there. Now these new mutations may spread, they may be found in other individuals, they may be lost by natural selection because it's bad. If it causes instant death, obviously it'll be lost very quickly. It may be lost from other forces, it may be lost just from a random event. That again, the individual with a new mutation gets hit by a truck when they're still young. Now, while they're present, these new mutations, these new variations will have a frequency in the population. And this is something that will be very important when we get into population genetics, that alleles are present at a frequency within populations. Now you can see this as an example. Here are allele frequencies of blood types in the United States, just as an illustration. About 24% of the alleles out there are A, 10% are B, 66% are O. Okay, so if you took just what's going on, they took just everybody's chromosome and just lined them up, you would count on average these numbers in the United States. Now these are the allele frequencies, but there's also an underlying genotype frequency, right? That a subset of these individuals have to be big A versus, well, it's obviously big A. AA versus AO, a subset are AB, a subset are OO, etc. So you can put all these things together, and these genotypes, the fraction of individuals that have a particular genotype, have some sort of frequency in the population, and that is what is studied in the field of population. We'll come back to that far more extensively in later videos. But again, all genetic variations started as these new mutations. Now, the tougher question is, how often do bad new mutations arise? Well, there have been estimates of this as well. The deleterious mutation rate is, again, fairly high. Now, if you look at chromosomal anomalies, things like just severe chromosomal disruptions, these happen quite a bit, and they probably have a very immediate effect on offspring and result in miscarriage. So as many of you know, on the order of about half of pregnancies are spontaneously aborted. It's estimated that maybe around half of those ones that are spontaneously aborted had some sort of genetic reason for it. They had some sort of chromosomal anomaly. They got too many copies of one, or things stuck together that weren't supposed to stick together, things like that. These are ones that you can't actually measure because it happens so frequently, there's no way to tell. But a lot of other bad mutations are far more subtle, right? They don't have such severe effects that they just make an embryo die, or they're masked because they're recessive. Well, current estimates suggest that on average, and this is true for humans as well, there are 1 to 2 bad new mutations per generation, per individual, right? So I'm sorry, but you're probably a bad mutant too. But I hope you'll come back nonetheless and watch the next video. Thank you very much. Hello, and welcome back to Introduction to Genetics and Evolution. In previous videos, we talked about mapping simple genetic traits. Those are traits whose variation is caused by alleles at a single gene. In the last set of videos, we talked about how most traits are not simple and various complications associated with genetic mapping. In this set of videos, we'll look at how we actually map complex traits. In this first video, we'll look at some of the principles used in mapping such complex traits. So let's get started. Now, as I mentioned before, most variable traits that people study tend to be controlled by many genes, not one gene. There's not a single cancer gene. There's not a single diabetes gene. But in fact, there are many genes that contribute to the likelihood you could get something like cancer, diabetes, schizophrenia, etc. Now, a couple of other traits that people have studied extensively are fruit size. This is actually one of the first traits, fruit size in tomatoes, one of the first traits that was nailed down to a single gene's effect. Now, there are many genes that contribute to it, but one of the genes was actually identified. Human height is definitely one that would be controlled by many genes. And a funny one that a lot of geneticists study is bristle number in Drosophila. So on the back of a fruit fly, you have all these little hairs. These are their bristles. And something a lot of people try to map as a nice quantitative sort of trait where there's a lot of variation among individuals are the number of bristles on the backs of flies. But obviously, a major focus, especially coming from the Human Genome Project, was understanding the genetic basis of disease. Now, in the last video, I used this fictional simplified example of 6 genes contributing to human height. And as you saw, each of these genes is contributing an effect. The A gene, B gene, C gene, D gene, etc. These are all contributing to this variation, and yet we see this bell curve, this normal distribution of height. So how do we go about mapping all these different genes? Again, there's not one height gene. There's a lot of different genes. Well, the concepts for doing this are very simple for mapping simple single gene traits. Okay? Nonetheless, as I mentioned, there are many genes contributing. Well, again, just like before, there's 2 general approaches. And we'll come back to these in subsequent videos. I want to emphasize in this video just some of the overlying principles. One of them is mapping the difference using crosses or pedigrees. This is exactly what we do with the simple traits. The other one is mapping variation within populations, and we'll talk about the Genome-Wide Association Study, often abbreviated as GWAS. I'd like to emphasize a few points before we get started. First, we often know the locations of the markers being used ahead of time. So if we're using something like, say, mapping in humans, we have a fully assembled human genome sequence. We have sites that we know are variable, these so-called SNPs, or single nucleotide polymorphisms. These have been identified from genome sequences. So we know where the markers are. However, we don't know the locations of the genes causing traits or diseases ahead of time. Okay, so we know where the markers are, but we don't know where the genes contributing to cancer are. We know there are genes that are contributing to cancer, but we haven't identified them ahead of time. The purpose of mapping is to find where they are relative to markers. So we're using their linkage to the markers to find these genes that are causing traits or diseases, to identify where in this vast genome sequence they are. Okay? Now, we map complex traits to things we call QTLs. That's short for quantitative trait locus, or plural quantitative trait loci. Now, these are places in the genome, or locus, with allelic variation that influence a phenotype. Now again, ahead of time, we don't know where these QTLs are. These QTLs are things that we infer from the data, from the mapping data. And at some level, you can think of them as almost a hypothesis, that we are inferring it's here, but it may not actually be right there. And we'll come back to this a little bit later on, how something that we map as a QTL may not actually be the location of the gene causing the trait. But nonetheless, we infer the approximate location from the association with marker genotypes. Now, very often, we detect many QTLs affecting single traits, so something like height, for example. But just as before, the fundamental underlying principle is we're looking for an association of marker genotype with phenotype. Okay? We're looking for that. And the reason for this is that markers that are near or linked to the genes causing different phenotypes should show some association with the phenotype. And I'll show you what I mean by this in just a moment. Now, if there are many genes, or if the effect is complicated, the association may not be very strong. So this isn't going to be as precise and as clear-cut as it was before, where we could say, oh, it's 1 centimorgan or 1% recombination away from this. This is going to be far less precise. But it's nonetheless possible to localize the location of these QTLs, and therefore the genes affecting traits. Let me give you an example. Let's imagine you're crossing some corn strains. So let's say you have some tall corn, which let's say is 5 feet tall, and some short corn, which we'll say is 1 foot tall. That would be anomalously short corn, but let's pretend that's okay. Now let's say the F1s, the offspring, are intermediate, so they're 3 feet on average. And they're pretty consistent about being 3 feet tall. Now we cross the F1s together to get an F2, and F2 is just a cross from F1 as the offspring from a pair of F1s. And let's say the F1s show this range from 1 feet to 5 feet, so they recapitulate the range seen in the parents. So we get something like a bell curve around 3 feet, something like this. Or here's 1 foot, here's 5 feet. We get a lot of 3 feet ones, some 4 feet ones, some 2 feet ones. Okay? Now, assuming the height variation is genetic, how many genes are involved? Is this likely to be something that is a single gene effect? The answer is really unlikely, right? There would have to be a lot of different alleles that are doing this. So probably not. It's much more likely to be something that's caused by many genes. Nonetheless, we know that this height difference is at least partially genetic, so there are genes affecting height. Okay? So let's say we got a genotype for 2 markers in the process of mapping this height difference among the corns. And let's say the capital letter ones are the ones from tall, as we use them here, A and Z. So let's say we look for an association between genotype and average phenotype that comes with that genotype. So here, individuals with markers homozygous for the big A marker are 3.5 feet tall. Individuals that are heterozygous for big A and little a, 3 feet tall. And little a's are 2.5 feet tall. Okay? Contrast, look at the Z marker. Big Z is 3 feet tall on average. Big Z is 3 feet tall. Little z is 3 feet tall. Now I'm saying this, I don't mean every single individual that's big Z, little z, and little z is 3 feet tall. But the average among individuals with that genotype at that marker is 3 feet tall. So is either marker associated with the phenotype? Do we see genotypes at these markers differentiating the phenotype? Well, obviously the answer is yes. In this particular example, Z is not associated with the phenotype, right? Because we don't see any difference among these genotypes in terms of the phenotype. But here, we do see a difference among the genotypes with the phenotype. Now let me ask you a different question. Why wasn't the association of the alleles with the A marker much more complete? Why weren't big A individuals 5 feet tall and little a individuals 1 foot tall? Because that's what we saw with the parental. Why didn't we see that? Well, the answer is all the things we talked about last time and one. So there's the possibility that there's multiple genes affecting the phenotype. This one's very likely to be true. It's possible that there's interactions among the genes or epistasis, right? All the things we talked about last time, all these complications. But an additional one that I want to factor in here, this is the essence of genetic mapping, is that there's been recombination between the marker genes, basically these A and Z genes, and the genes affecting the phenotype, so an actual gene affecting height. But there's some recombination between those two. And what happens then is this recombination between the marker and the genes affecting the phenotype weaken the association. If you had no recombination at all between the marker and the trait gene, then the marker genotype should very well predict the phenotype. Okay, this is especially true if it was a single gene trait, but this is somewhat true even if it's a trait with many genes. If you have a little bit of recombination, you weaken that association. It still may have some association, but it's weaker. And if there's lots of recombination, then there's no association. Again, this is the fundamental underlying principle of all genetic mapping. Now, let me ask you this question. How do we pinpoint gene locations in the middle of all this madness? Right? Well, at a very crude level, very crude level, we can just look for associations between marker alleles to phenotype, and we can say that there is a gene near there affecting that trait. That's not particularly satisfying. That would be like, you're trying to find somebody who's missing, and you pinpoint them like this. They are in this area of the state of North Carolina. Well, that's not very helpful. What we really want is fine localization. In this case, we want to be able to point at a particular spot in the genome, or using this example, a spot on a map, and say this person is in this building here. So what we want to do this is we want to look at increasing association of neighboring markers. We're going to look at a trajectory of association. We'll look at markers in a sequence, And as we get closer and closer to the gene with variation affecting the trait with these different markers, we should see stronger and stronger associations. Okay? Now, fine localization, as I mentioned, requires examining associations of multiple linked markers with the trait. Not completely linked, but markers that are very close together. And we'll look for a stronger association in some markers than others. Maybe you may be asking yourself, what do I mean by a stronger association? Well, a stronger association means there's a bigger difference between the average phenotypes associated with the genotypes. So remember, the Z marker I drew out before had no association. I'll actually draw that on here just as an illustration here. So the Z marker at 3 feet, 3 feet, 3 feet. This had no association whatsoever with the trait of interest, in this case being height of corn. We'll look at the A1, and we do see this difference. This is the one we saw before. Here's a B1. Well, if you look at this B1, there's actually a bigger difference between the average phenotype associated with the genotype. So let's assume that A and B are near each other. Let's say here's A, here's B, and let's say Z is very far away. Okay? If we see this, we say, okay, A has a weak association, B has a stronger association, right? Because we see this bigger average difference. We might infer that the gene causing this difference in height is probably closer to B than it is to A. Okay, so essentially we want to focus in near this marker B. Now again, association will be stronger if there's less recombination between a marker and the causative gene, or the QTL. So from this, we generate a prediction that if we look at many linked markers, we should be able to pinpoint the location of a QTL by where the association is strongest, or where we predict it to be strongest. Like we'll infer a continuing association going further along there. So to do this, we'll follow the trajectory of association strength to infer exactly where this QTL will be. Now we'll do this in the very next video. Thank you very much for your time. I hope this was helpful. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about the principles associated with mapping complex traits, and how we do this in both crosses and using genome-wide population studies. In this video, we'll elaborate on the previous one to look at actually what's done in genetic crosses to pinpoint the location of genes that that have allelic variation affecting some sort of trait. Now again, as I said at the very end of the last video, the ultimate prediction is that if we look at many linked markers, we should be able to pinpoint the location of a QTL by where the association is strongest. And we talked about exactly what that meant in that context. So what we'll do is we'll follow the trajectory of association strength to infer the location of the QTL. Now let me illustrate this with an example so you know what I mean, because trajectory of association strength may be a little bit difficult to understand. So let's use an example from the mapping cross. We'll stick with the same example of corn that we used last time. This was a cross where we crossed a tall strain to a short strain. The tall strain was 5 feet tall. Short strain was 1 foot tall. We got F1s that were intermediate. They were 3 feet tall. And then we crossed the F1s to each other and got a lot of variation where some individuals were 5 foot tall, some individuals were 1, and a lot of them were kind of in the middle. We got that sort of bell curve. Now what we're not trying to do is we're not trying to get recombination fractions this time. This is different from what we did in the context of simple genetic traits. It's much more complicated when something is complex, so we're not going to do that. What we're just going to be doing is looking at the association of genotype and phenotype, okay, just looking at that association, and we'll look and see how strong it is. Now remember, strength is defined by the average difference between the genotypes, okay? So what we'll do is we'll genotype 5 markers rather than 2 this time, and look at how they associate with height in these corn, okay? So let's dive in. So let's say you're looking at 5 close markers to the trait. These markers are about 2 centimorgans apart from each other. So the markers recombine from each other, the alleles of these markers recombine from each other at an average rate of about 2%. Now let's say we're going to put on our divine glasses where we now can see what the actual answer will be. So our divine glasses, we see that the height gene is next to C. Now we don't know that, as scientists we don't typically know that, but for this example, let's say that we do. Would we see an association of height with the E genotype? Okay, so we know the height gene is next to C. Would we see an association with the E genotype? Well, the E gene is only about 4 centimorgans away. So sure, of course we'd see an association with it. It's pretty darn close to that gene which is contributing to variation in height. Well, we might see something like this, where the E gene, if you're big E, if you're 3.5 feet tall. In this case, again, I'm using the case where all the capital letters are derived from the tall parent and the lowercase letters derived from the short parent. The big E ones are 3.5, the little e ones are 2.5. Okay, so again, we do predict some association between E and height because it's only about 4 centimorgans away. Well, what about D? Do we think there'll be any association between D and height? Well, obviously the answer is yes. Sure, but the important thing here is we see the genotypes at D are more strongly associated with height. So you see the difference here between big E, and little e was about 1 foot. Here, between big D, and little d, it's 1.4 feet. So more of the variation is explained by genotypes at D than by genotypes at E. And that's because the D marker is closer to the gene that's causing the height difference. So let's fill in the rest of these. There we go. So we see the same sort of thing here. What we see is as you get further away from the height gene, less variation is explained. So when you're 4 centimorgans out, you're explaining about 1 foot of difference in height. When you're a little bit closer, you're explaining 1.4. And when you're pretty much right on it, you're explaining almost 2 feet of the height difference. Okay, so the prediction is that we see a stronger association when we're looking at markers closest to the gene affecting the trait, which in this case is corn height. Now this has been done quite a bit, and this is often depicted using what's referred to as a Lod plot. So what they do is they use math, which we're not going to cover in this lecture, to translate this observed and expected associations to identify a likely position of the genes affecting the trait of interest. So in this case, this is looking at various traits associated with tomato, and pH here is very well explained. And you see, looking across chromosome 10, there's an area here where there's a lot of the variation in pH explained, and there's areas where it's not. Now, typically speaking with Lod plots, a high number means a more likely location for genes causing the effect. Often the cutoff of about 3 is used, and if you see a Lod score above about a 3, that means you see a statistically significant association. So what this would actually mean is, they looked at markers at all these different places, right? The markers aren't actually drawn on here, and this one right here had a very strong difference between individuals that were big P, and little p, in terms of the pH. This one had a weaker one, this one had a weaker one, etc. So we infer that a gene affecting pH is probably right around there. Okay, now it's an inference, it's not an exact pinpointing, but it's an inference of a likely location for it. So how does this differ from when we were mapping simple traits? Well, the big difference now is that we're looking at markers in multiple regions of the genome associated with the trait, and a lot of them are explaining it. So this picture here is from a map of QTLs that are associated with Drosophila mating success. As you can see, there's a lot of these little peaks. This is the same sort of Lod plot as I just showed you. So here, this is the X chromosome, 2nd chromosome, 3rd chromosome. There's 3 different places on the 3rd chromosome, at least, maybe a 4th, that are associated with variation in mating success. These little triangles that are a little hard to see in the very bottom, those are actually the locations of the markers. So what we can infer is the association of this marker right here is bigger with mating success than the association of these markers here, which is effectively negligible. Okay, same sort of thing here. Big association with this marker, negligible with that one, relatively speaking. So you're inferring from this trajectory where the likely positions are of these genes affecting the trait. Now let me give you one to try. Infer the location of the QTL by the trajectories of association here. This is basically the same principle as before, but what would you predict in this case? Here we have an association with A, B, C, and D. Hopefully that wasn't too difficult for you. What you saw is that A and D had weaker associations than B and C. B and C had similar associations. So probably, as you're looking at this trajectory of association strength, we see it getting stronger this way, we see it getting stronger this way. So you'd probably infer that the gene causing this height difference is probably somewhere between B and C. That's the difference from what we did before, right? In this case, it wasn't right at C, but it was actually between 2 of them. So you might make a Lod plot that looked something along the lines of this. Now, I mentioned to you before, these things are hypotheses, and there are dangers. You can actually be misled. Now, let's imagine that we looked at another gene right in the middle, another marker. Let's say it's the marker T. What we expect is T should have a stronger association with this trait than B or C. But what if we do that and we see this? Huh. Well, what does that imply? Well, in this case, adding the T marker in there, we see the T has a weaker association than B or C. So rather than having one single large mapped effect, it looks like maybe we're potentially mapping two different genes that have allelic variation contributing to this. So maybe there's one near B and one near C rather than a single one near the middle. This happens quite a bit in the course of genetic mapping, something which looks like one strong association, therefore a single gene with a large effect. So you map a little bit more finely, and ends up being, actually there's a couple of different genes in that same area that just happen to be a little bit close to each other. So we don't know what makes the QTL effect. We see these effects in mapping, and these are inferences of the locations of a factor or factors that are contributing to this variation. There may be multiple genes that are close together, or there may be a single gene. But the reason I stress this, and this is very important, the QTL mapping, whether it's by cross or pedigree or by population association study, is not conclusive. It is a hypothesis of where one or more genes are affecting the trait. And ultimately, you have to go in and do other techniques to confirm that is truly where there is a gene that has allelic variation contributing to this trait. Hope that was helpful. Thank you for joining us.